So we're basically trying to go from the least mysterious system to more mysterious systems. We are a very mysterious system, and we need to know what are all the pieces, on the way. So if you liked this ground truth model idea, but you thought, ooh, going for, ChatGPT or GPT-2 was a little much and it's not close enough to biology, this series of experiments might be a little bit more interesting. So we want to start with a known ground truth neural system, understand in silico what our system is doing, but then build up using neurological models. So like a known system, maybe neural array, so not even an organism yet, but a neural array interacts with electrodes in a virtual environment. We know everything about it pretty much, and then could do experiments on it in vivo, as well as start adapting our model to be able to act like it. So basically ever increasingly complex systems that we can then emulate and find what those unknowns are. What are the things that we don't know?
What this also includes is that you develop your metrics along the way to determine exactly how you say that your system is actually similar to the thing that you originally had. It also involves adding to just the system dynamics plasticity of the system. And then ultimately it allows you to compare methods. So for example, you've got all of your data from your known ground truth system that is functional, and you can use the functional data to come up with your emulation. But you can also, of course, using the pretty printer, pretty much, the imaging version of it, and try to get to the same thing, and prove out that indeed you can use morphology or tagged morphology to get to the same result. And then work your way up. I think maybe I'll hand it off again.
We wrote a lot here because there's like last minute. So I think the increasing complexity, this is very scalable. We start with as little mystery as possible, scale up to neural arrays, organoids, small organisms, and then more complex organisms. The general approach is to design an iterative feedback loop to test these systems, gaining those metrics, finding the challenges along the way, identify solutions, and there is actually a presentation linked at the bottom that goes into more of the justifications. So limitations on how these, I think we might have misunderstood this question. So this isn't how it's being done right now. System models have no ground truth, so they don't know how well they're really working. It's sort of guessing behavioral from the top.
Yeah, so I think part of it is that what this really is, it's a proof of concept that you can upload, let's just say, something. Whether it's just a few neurons in culture or a very tiny animal with 200 neurons or something like that. You're able to show and prove, because you have the metrics to show how it's similar, that you can actually upload something. And if you compare with today's approaches of attempting to upload something, well, these models, like for example, if you're trying to build a hippocampal neuroprosthesis, they don't have the full ground truth for theirs, so they can't tell quite. Or if you have the work doing visual neurons in Drosophila, you've got, some of a system, you've got the EM data, and then you have a bunch of other information that you're taking from other experiments you're using as assumptions to build in to try to create a visual neuron, recreate at least a few of the neurons. You never have quite the ground truth the way that you could in this case. So then it's very hard to tell if you've really accomplished what you set out to do. So the main part of this is just to prove out what is your success criterion, what are your metrics for it, how can you show that you've actually uploaded something, and then can you show how you go from there to the next step? Can you make it really progressively obvious that you're identifying the problems, coming up with the solutions, finding out systematic ways to make it efficient to get to your ultimate goal? A very sort of systematic approach, but beginning with knowing exactly what you're trying to do and how you measure that you've actually achieved what you're trying to do.
Are we done?
Yeah, so we're wanting to do a lot of in vivo testing as we go, as these systems are training. And then after, if we can, slice and scan and see if what we can take yet again from that, and then how close we got to the in vivo model, how close we got to original thing.
That's a really dumb question. Like if you have metal spikes going through the neurons, and then you kill them and fix them, and you want to expand them, as you probably would, how do you imagine registering where the spikes were or even preventing like that process from causing distortions as it expands?
Okay, so there are actually two different things to answer here. First of all, in the very first example, you don't put spikes through the neurons, because when you're building cultured neurons on, say, an array, those neurons are growing in your array. You're not putting spikes through your neurons.
Around metal.
Right, that's
Yeah.
the first thing. The other part is that often if you have, say, any kind of brain structure that you've had electrodes in, even if it's not, like, say, on an array, but like, say, just a brain from an animal and you put electrodes in, frequently people will just leave the electrodes in and they'll fix it and then they'll just kind of carve through it and see them in there. You can actually find the trace of the electrode. Or if you've removed the electrode, you can find traces of where the electrode was.
So you don't think it'd be too bad to register?
To identify where they were, no, but you could also use a more low fidelity approach to figure out where your electrodes are. Like you can put it in a, you know, in a CAT scan or something like that and just see where the electrodes are.
Also have an expandable array.
That's a good idea, yep.
Cool. So I'm not totally sure where we are on here. I'm going to suggest if people are interested, they can read through more justifications. But as far as how the project contributes to AI safety, it contributes by giving humans, you know, ability to look forward, and you know, the whole WBE speed up portion of it, basically. How would it contribute into the general WBE roadmap? I mean, it is part of the pipeline of getting from simple systems to human systems. So it is a big part of that. How much would it cost? It really depends. So if we wanted to do the whole biolab, have everything under control, you know, we're definitely talking in upwards of $1 million to set up and run that lab. But if we can collaborate with a biolab to do all of this neural work, like the neuroscience side of it could be done in basically half a million. So that is approximate cost. And proof of concept, first in silico and in culture, could be doable within a year. Process of discovering methods, going from step to step, takes a little bit more time. And as we run into these unknowns, you know, we can start making better and better predictions.
Yeah, but it also depends on your funding, I guess, right? Because
Well,
for the next part, because as you're building your tool set, and you've got your in silico tool set, and your how do you work with biology tool set, and your metrics that you're building up, you can parallelize many of the other experiments that follow as you go from step to step. And then, of course, you have to constantly keep on improving your tools. And
And this is an area where I think having feedback would be awesome, too, just so that we do get better and we make sure we're building a really good project plan. As far as completeness, this is by... Okay, we're almost done. It is by nature iterative and scalable, so each step is testable, generates metrics for the next step. It's not all or nothing. We learn every single step of the way, and it will generate methods to get better for each of the future steps. As far as what we would do with the small grant, try to find the bigger money is a big part of it. Refine, yeah. Yeah, and you know, maybe do some of that biolab research because we have a little ways to go on that. So setting up next steps, basically.
Thanks. We have time for maybe one comment or question?
How do you grow cognitively with this?
I'm sorry, what?
Because Bubba said it helps people grow cognitively and stuff.
so the part about like how does it help in safe AI, all of the arguments in there, maybe misunderstood exactly right, it was basically about, yes, we're working on whole brain emulation, so when you get whole brain emulation, how does that help you with AI safety?
Okay.
Yeah.
So it's in that order.
Yeah, sorry.
All right.
Okay.
Well, thank you guys.